Welcome all of you to today's webinar. This is the last one out in the series of in total 4 webinars that we have organized during the fall, covering different phases of the drug discovery process with a special focus on how the CETSA method can be applied all along the value chain to, for example, strengthen target validation or to better understand mode of action or for biomarker discovery. So the title of today is Biomarker Discovery with CETSA Explore, focus on the protein status. And this presenter is Daniel Martinez Molina, our CSO of Pelago. And a little bit of Daniel's background is that he started his career at Karolinska Institute, where he developed methods for optimizing membrane protein expression and stability for structural investigations. And he solved the first high-resolution structure of a human membrane protein. After a postdoc period at Stockholm University, he was recruited back to Karolinska Institute to specifically develop the CETSA method. And the first proof of concept paper came out in Science 2013. And Pelago Bioscience was founded at that time. So here we are 8 years later, and I'm very happy to have Daniel on stage today to talk about this topic. But before I leave over to Daniel, I just want to say that this presentation will be recorded. So please stay muted during the presentation and also have your camera off. And also please write questions in the chat if you're eager and have questions all during the presentation. And then we'll have a Q&A afterwards so we can discuss and bring up the questions you have. So thank you very much, and the stage is yours, Daniel.
Thank you. So I guess I'm not muted anymore. Yeah, so thanks for the introduction there, Helena. I'm, as always, happy to talk CETSA and how we apply it here at Pelago Bioscience. Today, I'm going to shine some light on a project that we've been running for a couple of years now. We call it the Target Engagement Atlas. And some of the findings there that could lead towards the biomarker discovery field. So first of all, I'm proud to work at Pelago. It's the home of CETSA. We serve as a specialized CRO focusing on biologically relevant assays to prove target engagement, to identify targets and their modes of actions. We want to be the partner of choice in the development of new medicines, and we work hard to make sure that both we and our customers get the most out of our data. The Pelago team, including our expert R&D team, are located in Stockholm, Sweden. First, I wanted to dissect my title a bit, just to give you a hint of what I will be covering today. First of all, CETSA, I already said the word 4 times. It's my favorite topic, and I will give you a short intro to the method and then carry on to show data. Then biomarkers in this context and for the remainder of the talk, basically, it's any kind of signal, protein signal or protein behavior that in a reproducible fashion can correlate with target engagement and the subsequent cellular responses. The ideal biomarker for keeping track of the efficacy of a drug is probably the target protein itself, if the target and the drug mechanism has been properly evaluated, of course. The drug target is, in fact, the closest in proximity if you want to grip on efficacy. And the phenotypic effect is then down the line and can be diluted, so to say. However, very few drugs hit just one protein. So for a full understanding of a mechanism of a drug, you probably need to study more than just the target itself, just to get a feeling of unwanted side effects and so on. In the first part of the talk, I hope to convey that CETSA is indeed a tool for finding biomarkers in the preclinical space, I should say. And here it's mainly focused on safety biomarkers and monitoring of target engagement that I'm referring to. We have only limited experience with clinical biomarker work, but we are gearing up to do more of that. And if I have time at the end of the talk, I'm going to go through some of our recent data. Finally, focus on protein states. What I want to do here is, in principle, just push protein biomarkers in general. Genetic and transcriptomics-based biomarkers, they are good, but not for everything. Transcript levels don't always correlate with protein amounts. And also, to be fair, protein amounts don't always correlate with functionality of the protein. And so I hope that with CETSA, we can also have a grip on the states of the proteins. So with CETSA, and that's what I'm going to get to at the end, we believe that we can distinguish proteins in different contexts by measuring the melt temperature in the cell, melting behavior, and the response patterns to specific drugs. At the end of the talk, I will show you some of the fresh data that we have that could support this. So CETSA, it stands for Cellular Target Engagement Assay. And by the name, you understand that this is a thermal shift assay. So it's a biophysical method that we carry out in the living cell. We use the thermodynamic phenomenon that is when a complex forms, there will be a net difference in the stability of either one or the other or both partners of such a complex. The method works without any labels or modifications. So it's quite enabling technology that can be used to from bench to bedside in principle. In the hypothetical example on the left here, we measure the stability of a blue protein. When a small drug binds to it, it turns cyan and is then more stabilized. And this is in principle also what we studied in the first proof of principle paper on the right. So the method works by subjecting cells to a heat pulse, and then that heat pulse need to be fine-tuned enough to be able to catch the difference between proteins in their different binding states of the compound. We can work qualitatively, like can you see my point? Can you see my pointer here? I hope so. Yeah, so we can work qualitatively like this, say that there is a shift, or we can settle on a temperature where we have a good resolution, and we can then screen in a quantitative fashion by applying a range of doses like these examples here. To many, it's not a surprise that CETSA also detects changes in protein-protein interactions, and because of this and the fact that we work in living cells, we can observe cellular responses to a drug, not just study the primary target and the productive binding of a drug. So a lot in the cell is about protein complexes, and we see these drawings all the time in scientific publications. Some of them are more detailed than others, both from an artistic but also a scientific point of view. But what they all want or have in common is that they want to explain how a specific corner of the cell function. It's obvious from these that we do understand that very few proteins act on their own. We see in the drawings that they are either part of a complex, they're leaving one, or forming one as a consequence of a signal that is usually the main theme of that particular publication. Also, these protein-protein interactions, moving from one form, a dimer, into monomers, for example, has consequences on the thermostability of the involved proteins. That's what I said in the previous slide in principle. So you have the interaction surfaces between two proteins. They're usually quite big, and they can supply ample and specific interaction surfaces, which either cost a lot of energy to break or offer a more stable conformation when it's formed. I'm going to come back to this reasoning later on when we focus more on the CETSA data produced with mass spec readout. In the mass spec version of CETSA protocol, we can easily follow thermostability changes of 6,000 to 7,000 proteins in a typical experiment. But first, the basic steps of the CETSA protocol are, and I don't think I will dwell too much on this, it's 4 steps. You see the first 3 here. You treat your cells with a compound of interest. You heat your cells after a certain incubation time. Then you separate the protein that has precipitated by this heat pulse from those proteins that hasn't. And according to thermal shift assay theory, there will be more of protein X or Y compared to control sample if they were stabilized by the treatment, and vice versa. So less protein in the soluble form at each temperature if it has been destabilized. And since this is a protein-centric method, we can in principle apply any fit for purpose protein detection technique at the end of this protocol. We have divided it into different approaches. So you have either the targeted one that we call navigate. These are usually antibody-based, so you have to know what target you want to study. And they come in different throughput formats. So we can do Western blot, so we can run 384 well plates to screen large compound libraries. These, however, they have been covered in previous webinars. But I just wanted to show one example where we used a JES setup from Biotechne when we worked with the protein STING. And as you can see here, STING is stabilized upon addition of the compound diabsi to the cells, whereas the complex partner binding here in purple is untouched. So with JES, we could easily multiplex this, and as you can see, the bands are crisp, and the size-wise, they don't overlap. To me, this data was quite interesting because I wouldn't have been surprised if TBK1 was also affected by the treatment, maybe a D-stab or something. But it may be that the abscess is able to bind STING without affecting the TBK1 interaction. Or alternatively, that the melting behavior we see of TBK1 here and STING is from their monomeric forms, i.e. in this cell system, they do not form a dimer. However, of course, it's good if you have a target protein that is part of a complex to make sure that your compound is indeed hitting the right protein and not another one in the complex, as it could lead to the same phenotypic effect in the end. So back to this, and apart from the targeted navigate assay, we also have the Explore formats, which are more of a discovery tool instead. We use mass spectrometry for detection, and similar to the targeted assays, we can also look at specific proteins, obviously, that we think or hope bind to the compound of interest. But more importantly, since we cover 6,000 or 7,000 proteins per experiment, and because we use unmodified compounds in cells, we think that it's a very powerful, unbiased way of looking at cellular perturbations following drug treatment and for discovering biomarkers, both in the preclinical stages of drug discovery, but hopefully also in a clinical setting. We call this CETSA MS as well, but you may have heard of the method as TPP, thermal proteome profiling, before. The TPP data, what does the data look like then? On the left, you do not have TPP data. You actually have data that is produced with an alpha LISA kit on ERK. So you see the stepwise stabilization of ERK as a consequence of increasing doses of ravoxertinib. But on the right, you see the data set of how it would look in MS in our most complex protocol, which was the 2D TPP. Here you basically have a melt curve at each concentration, or a concentration response curve at every temperature. This was a very I shouldn't say very suboptimal, but it was a suboptimal way to collect data, as it required lots of reagents, lots of mass spec time, and it was also hard to apply statistics during the analysis steps. So instead, we developed a simplified version of the CETSA MS protocol. We refer to this as compressed CETSA, but you may also have heard of CETSA 1-pot or PISA. They are in principle two alternative names, so it's the same method, but it's just developed from the original CETSA protocol in different labs. In principle, we run the assay as normal, so full melt curves and the possibility to run different concentrations. We then pool the samples after the heating. So in the sample, before the centrifugation step, that's the separation step, we have the, you could probably call it the physical integral of the area between these curves. So basically something like this. And this way, we can then reduce the sample sizes, the time on the machine, etc. And in some cases, we do run full dose response curves this way. But in the remainder of the talk, I will mainly focus on compressed CETSA data from single high concentration experiments. So principle like this one. The data is usually best visualized as volcano plots like this on the right, where you have the numericals depicting, in principle, the area between the curves on the x-axis. So that's the fold change and the height of the green bar here, so the difference between two control. Then on the y-axis, you have the significance of the data. And this particular example shows the 30 micromolar concentration point of methotrexate. You see the well-known target protein DHFR, as well as several other proteins that are involved in the folate and thymidine metabolism. And as such, this is a good example of how CETSA can pick up hits that correlate well with the compound's known mode of action. And most proteins here highlight that we're looking at the binding, the direct binding of the compound, not only to the intended target, but also off targets that have an affinity to it. But you also see some downstream events of the target. So with that, I wanted to talk more about the Target Engagement Atlas. And this initiative started at Pelago back in 2018 when we applied for and received funding from the innovation agency Vinnova to establish a database of CETSA profiles for compounds with a known hepatotoxic side effect. This initiative grew quite rapidly when we realized how much information we obtained about the molecular mode of action of the compounds. And today we have profiled a bit more than 400 compounds, and many of them in multiple cell lines, as I will show you later on. The initial idea, however, of finding biomarkers for liver tox, that's still ongoing. It required more data and better defined types of liver tox than we had anticipated at first. And just a quick intro to how we produce the data. We run relatively short incubations of 1 hour. We do not want expression level changes. We simply want to monitor the initial binding events. We run one single and saturating concentration. We do biological triplicates, and we automate as much as possible. Each compound then produces its own CETSA fingerprint or MS profile, like the one you see above, again, for methotrexate. And then these type of volcanoes, they exist for every compound that we try. And that way, it's possible to apply some advanced statistics and start looking for patterns in the data. Like this. This slide is getting a bit old, but it still serves the purpose of summarizing the results from the first year. This is a t-SNE plot of the first 190 something compounds that we ran in HepG2 cells. This is then an unsupervised non-linear reduction model that makes it much easier for us to visualize high-dimensional data. In principle, and again, I hope you see my pointer, compounds that lie close to each other have a similar CETSA profile or volcano plot, and the further away they are from each other, the less similar they are. So, for example, up here, you have the mTOR inhibitors that act similarly on the cell. To the left, you have kinase inhibitors, but you also see kinase inhibitors scattered throughout the whole plot, as they are often relatively specific in hitting clusters of kinases, or they have different or similar cellular downstream reactions. Down here, you see tubulin inhibitors, but you can also see colchicine here. This is because colchicine is an alkaloid, and vincristine, vinblastine, and vinorelbine, they are also alkaloids, but vinca alkaloids, so they have that part in common. So that's likely why they stack together. Then you have docetaxel, which has the common theme with the vinca alkaloids, that they bind it binds the tubulins. And on the right here, you have a cluster of antibiotics where this single antiviral raltegravir has nested in and shows some, I would say, quite interesting similarities to the antibiotics. We could also highlight the Enzyme group of compounds. We see them stack into 3 main clusters, and it could be interesting to understand why. So we then focus in on the underlying data that causes this clustering. So if we look, for example, at Diclofenac and lumiracoxib up here, they stack on top of each other, and so does piroxicam, meloxicam, and droxicam. And we then look here, and up on top here you have the diclofenac and lumiracoxib, and they share this cluster of proteins that respond to the treatment, and then you have the proteins on the x-axis. The same for droxypyro and meloxicam. They share similarities in this region, maybe some over here, and also the SADH2 here, which is the same as PTGR3. Also, importantly, we cannot always access the direct target of a drug. So for example, celecoxib, for example, we don't see COX-2 in the data, but we often see the downstream events, pretty much like ripples on the surface when you throw stones or other things in the water. And so again, the direct and primary target would be the best biomarker to signal if a compounded drug has reached its target, but we can also use other proteins as downstream biomarkers to signal for an unwanted effect, unwanted side effects, or simply use them as those shifts as proxies that a compound is present in the cell. So at this stage, it's quite obvious that we are not just looking at chemistry-driven binding profiles, we're also looking at biology-driven cellular responses. And it's not always that we understand what's happening in the cell. We simply see that something is happening, but we can't explain the biology. And this is fine, even though it's of course rewarding to understand why something happens, not just that it happens. One example where we think we were able to connect the dots was when we profiled 3 different CDK4 and 6 inhibitors. We saw that ribo, palbo, and abemaciclib had some similarities, but they also had differences. Similarities in that they all hit CDK6. CDK4, however, over here is not that impressed by palbociclib. CDK4 is found somewhere here in the cloud of thousands of other proteins that do not respond to ribociclib. We see they also have CAM-K2 delta protein responding in common. But one thing that drew our attention was how abemaciclib showed similarities to a CDK9 inhibitor that we had profiled for publication purposes together with AstraZeneca in the past. It also destabilized a protein with a funny name, FOXK1. And with our database, we could then easily find that compounds that cause destabilization of FOXK1 also induces a stability increase in the GSK3 proteins. There was a clear correlation. That's the one on top here in the scatter plot. So you can see that there are several compounds in our database that has the correlation between GSK3s and FOXK1. And in the CDK9 inhibitor project down here with AstraZeneca, we show with those response curves that they seem to go hand in hand concentration-wise. So you see CDK9 being the primary target for this particular compound, stabilized or was stabilized already here at nanomolar concentrations. Then it was followed by the GSK proteins. And you see the distinction between GSK3 alpha and beta, but you also see the protein GSKIP, which is the abbreviation for GSK3 beta interacting protein, which is known to bridge GSK3 beta to active protein kinase A to enhance Wnt signaling. Now this GSK3 FOXK1 correlation, that was quite easy for us to find by eye, and so was the biological explanation. Simply we just did a PubMed search, and then this paper in Molecular Cell showed up, and the authors there, they drew yet another of these nice cartoons for us. So what we believe is happening is that the binding and inhibition of GSK3 with these compounds, they cause an effect that is similar to the cellular mTOR mediated suppression of FOXK1 phosphorylation by GSK3. So the drug binds to GSK3, inhibits its phosphorylation activities, and causes FOXK1 to reside in the nucleus in its hypophosphorylated form. And then this is obviously then compared to the control sample in which it's phosphorylated and it binds to these 1433 proteins. So what we detect here is then a less stable nuclear form of the protein. Now, it's not always this easy to find correlations and explanations in the data, and we believe we probably have dozens of similar examples in our database that we cannot find by eye or nor explain with CETSA alone. So we are obviously eager to talk to others that could help us with this. The results I've shown you so far, they have been from one single cell line, HepG2s, which is a hepatocellular carcinoma. But we have also expanded the atlas to other cell lines. We run K562, which is a myeloid leukemia cell line, and U87MGs, which is the primary glioblastoma cell line. And this way, we believe we have a fairly broad repertoire of cell type backgrounds. And we were curious how these differences in biology that are represented there translate to target engagement profile and subsequent responses in these different cellular backgrounds. Again, we're still just looking at 1 hour incubation. So the cellular responses that I'm referring to, they are probably just some kind of springboard towards a less well-resolved phenotypic effect. And what we see so far in this part of the project, and I'm probably going to be a bit unspecific here, and that's simply because we haven't gotten that far in the analysis yet. So you're seeing quite new and quite new data. But what we see is that among the compounds that we've assayed in all cell lines, it's about 100, is that in general they overlap quite well for the primary target, if it's there. But then there's a lot of other things that happen as well that can be very cell line specific. Now, this slide is quite busy, but it's a good summary of the data. You see a large number of compounds here and here. Yeah, a large number of compounds, I should say, where the proteome behaves similarly, sometimes close to identical, but there are examples where this is not the case. And the most obvious ones, they are those that then cluster on or around the X or Y axis in the plots here. For example, you can see that this protein, ZBTB21, is shifted to the same extent in K562 and HepG2s by procarbazine. But when comparing U87 MG with K562, you see that there's no correlation. There's no response on ZBTB21 with procarbazine. The same is true for superoxide dismutase 2, SOD2, which is affected by both lumiracoxib and ruxolitinib treatment in K562, but they are not touched by in U87 MGs nor HepG2 cells. Similarly, you also see that imparsa has different effects in K562 compared to the other cell lines. And the more specific sample that we have spent more time on is the SARC family of kinases. And this family is composed of SARC itself. You also have YES, just one, FIN, LYN, and a few more. And here is one example where we believe that CETSA can pick up specific cellular and protein activation states that makes the protein shift differently between cell lines. And this is another one of those busy slides, but I hope I can walk you through it. So what we're looking at here is the correlation between how different compounds, those are the dots in the graphs or graph, affect either SRC itself, SARC, on the y-axis, or the SARC family member YES1 along the x-axis. And what you're looking at here is the log 2-fold changes. The different colors of the dots, they denote the cell type. So you should have 3 dots per compound, ideally, if it's covered in all 3 cell lines. And you can see by the line fits here that there is a correlation between the responses in HepG2, that's the red one, between these 2 proteins. And you can also see it for U87MG in green here, that there is a better correlation than it is for the blue line, where there is almost no there is no correlation between these proteins and their response patterns. And this indicates that these two proteins, although they are in the same family, have less similarities in their biological effect or availability to these compounds than in the other cell lines. And this is also obvious from the patterns. You can see from the red and the green dots that both these proteins are destabilized by the addition of all the compounds except for ponatinib and neratinib and HepG2s. But this general destab is what we had expected when we started this, because usually when Sark family proteins are treated with compound, they become destabilized. So this was one of the things that we picked up in our previous runs, but then we had only run HepG2s. And now when we added K562, we also saw that there was also a number of compounds that did not have a destabilizing effect in K562. It was actually either stabilized or not shifted at all. And you can see this for several compounds here: roxolitinib, avapritinib, vandetanib, lenvatinib, and crizotinib, for example. But you also find that those compounds that do cause a destabilization of Sark in K562 do so, and that's ripretinib and nintedanib, for example. They do so, but to a much lesser extent than in HepG2s or U87 MGs. And you can just draw these in this visualization. And these triangles, they also point to the fact that this protein, yes, is also affected to a lesser extent by nintedanib and ripretinib in the cell line U87MG than it is in K562 and HepG2s, respectively. So it's another indication of cellular preferences, if you wish. But anyway, back to Sark. Despite that many of these compounds are de novo Sark family inhibitors, or at least known tyrosine kinase inhibitors, the only compound that has an effect on Sark that is comparable to the other cell lines, in K562, that's actually dasatinib, which you have here. Now, we don't know why this is, but dasatinib is known as a Sark inhibitor, specific Sark inhibitor, not Sark family inhibitor, with a clear niche in the treatment of leukemias that test positive for the Philadelphia chromosome bearing the fusion protein BCR-ABL, which also happens to be the source tumor for this K562 cell line. So we have the biology here that also in the clinic seems to be very sensitive to the dasatinib treatment. So it may be that the dasatinib is so unique because of the good match of the biology of the Philadelphia positive cell types that is not represented elsewhere. We also, just as a control, we compared the protein fetch, ferrochelatase, which is frequently hit by kinase inhibitors. So we compared it to the yes-1 response. In principle, this is the same kind of plot as this one, but with fetch instead. And you can see, and that was expected, that there was no correlation between these two proteins. So we can't really see any clustering of compounds based on cell type, which then strengthens the previous data that there's no correlation. So that's why we hypothesize that the Sark family of kinases, they do exhibit different protein availability or states in different cell lines. And as you've seen, that makes them respond differently to drug treatment. I also included this PARP example showing the well-known PARP inhibitor Imparsa or olaparib in this slide, and its effect on PARP1 in those three cell lines. As you can see, you have the shift size on the x-axis here, and on the y-axis downwards in K562 versus HepG2 here, for example. You can see that PARP1 is stabilized to a great extent in HepG2s, but there is almost no effect of olaparib treatment in K562 or U87MG. So it doesn't really let go of the x-axis here. At the same time, the DNA strand breaker bleomycin causes massive destabilization of PARP1 in K562, as you can see here, and also in U87, but it almost doesn't affect PARP1 in HepG2 cells. So this is interesting, since bleomycin has a very high correlation between all these three cell lines with regards to its other cellular effects that we've measured with CETSA. I'm not showing that slide now, but there is a high correlation. And so PARP1 really stands out here. And it should also probably be noted that what we're looking at here is probably not the true destabilization. It's more likely that it's PARP degradation. That's a well-known inactivation process in systems where DNA damage is extensive, which is basically what bleomycin is causing. However, that doesn't seem to be the effect in the HepG2 cells, which has then this kind of reversed response pattern to these drugs. So very different cell line activities here. And also here, we do not have the full explanation to the cell discrepancies, but we do believe that applying this kind of CETSA MS screening to a standard set of cell lines could be useful for tox profiling or tissue specificities, or as well as drug repurposing, and probably general biomarker discovery would summarize that neatly. And that actually brings me to a summary slide. It's a summary slide of this part, which is the preclinical part. So we can say that CETSA monitors target engagement, off-target binding, and detects cellular downstream events. We can use CETSA fingerprints to identify similarities and differences in the mode of action. We keep the compound incubation time short, so below 1 hour, to make sure that we're looking at stability changes and not the expression level changes. The cells, they seem to have different response patterns to drug treatment, and that can have implications in off-target profiling, tissue specificity, animal model triaging, and biomarker discovery. And we also see that expression levels don't correlate with protein responses. I didn't point that out specifically, but for many of those proteins, we do see the same expression levels, more or less. So we at least detect the same number of peptides, and the data is equally strong, and so on. So we believe that it's not all about expression levels. Let's see, we have I don't know if we have time for this, but maybe I could spend a few minutes on our ongoing Prebiom project. It's an EU-funded collaboration between us and Oncolines in the Netherlands and then Pangaea in Spain. And the aim is to set up a biomarker platform for evaluating new immunomodulatory drugs. So at Pelago, we obviously focus at the target engagement part of this. And then Oncolines, or NTRC as they were called before, they keep track of the immune markers. Pangaea, they are the experts on genomic biomarkers. So we start in lung cancer, and we obtain samples from the Rosell Oncology Institute in Barcelona. And we look at PD-1 targeting antibodies, and that's plural to highlight differences in binding patterns between those on the market, and in order to find biomarkers that could be correlated to a clinical outcome. We obtain samples from two sources, either real patient samples from Spain, as I said, but also we do work with healthy donor material from Karolinska Hospital that we use to evaluate buffers and so on the protocol. We do not use the precious samples for that. And what we do is that we harvest the PBMCs, and for the non-deceased donor samples, we need to activate for PD-1 expression. And the first question we have to ask is, can we see PD-1 melt at all? It's a membrane protein, so it's not the easiest. It's very shy on the mass spectrometers, so we're approaching this using Western blots. Secondly, can we see it shift when we know that the antibody is there? And then I should probably say that the curve here is a random melt shift curve that I picked from the literature. It's not PD-1. So this is the targeted assay where we run the melt curves, and in the mass spec, we instead run compressed CETSA or PISA as more of a fishing expedition to see if we can pick up any other intracellular response patterns that differ between patients. The reason for the melt curve analysis is that we believe that the melting behavior of the protein can tell us something about the protein context. We say that, for example, PD-1 surrounding is important. So if PD-1 is in a complex, a responsive conformation, etc., that would be different, give a difference in the melting temperature. So obviously we can't tell simply by looking at the melt curve if PD-1 is in a complex or not, but we can look at the melting temperature of the protein if when it's in an untreated native state, to see if it differs between donors. Like this. First of all, this is one of the first Western blots of PD-1. They are a bit messier than we are used to when we work with cell lines, but we do still see the effect of pembrolizumab, one of the antibodies that are used in the clinic, when we add it. So you see, compared to control, we see a quite clear destabilization. And when we repeat this for 8 different donors, we see something interesting. This is obviously preliminary data, but this could confirm what I said before, that melt curves may signal what state a particular protein is in. So you can see, if you look at the non-red curves, and again, this is a bit messy, that there is a large spread of the TMs, and also the shape and steepness of the melt curves. However, when we feed the cells pembrolizumab, it seems that PD-1 assumes a conformation that is uniform, or more uniform at least, between the donors. That the binding of pembrolizumab sort of pulls PD-1 out of the original context, and the melting behavior then becomes much more uniform, actually giving a rather well-defined melting temperature of about 49 degrees here. Possibly, this could mean that different starting conformations of PD-1 could lead to different intracellular responses, since you force the protein out of different complexes, for example. It could also be that certain conformational starting points require different amount of pembrolizumab in order to adopt this conformation, melt curve shape or conformation. In this particular experiment, we tried only one high dose, so we don't see it in a quantitative way, if you compare it to what I said before in the introduction. Now, so if this holds all the way, it would suggest the monitoring of the cellular state of PD-1 using CETSA prior to treatment or during treatment, and not just in the expression levels, could be a useful way to set the dose. Obviously, we need to run this many more times and include more controls and so on before we say something definite, but this is so far an observation. I can't say anything about the MS assay that we run in parallel, because we don't have the data so far, but we're expecting it. So in conclusion to this, and I'm rushing through it, is that the melting profile of the target protein without any compound addition at all, as assayed from donors, may tell if the protein is in a receptive or non-receptive state, and ex vivo dosing may indicate how much of a compound would be needed to obtain full target engagement prior to the real dosing. In the mass spec experiments, we may find non-target biomarkers, so downstream events and so on, or simply we could explain any differences in the cellular responses. So finally, I'm just going to spend 1 minute on this slide. In the literature out there, there are now several examples where CETSA has been used on clinical samples or in projects to find biomarkers. On the left, this research team used CETSA on glutaminase as one of those targeted biomarkers of glutaminase binding. Again, the target protein is the closest one to the action. They show that this assay detects the binding of CB839 or telaglenastat to glutaminase in platelets of patient samples collected while receiving this drug in a clinical trial, so they could keep track of the dosing and the efficacy. Another example has been around for some time now, since 2018. That's from Martinez. Here he used CETSA MS to identify a biomarker, ECM29, and its predictive power for palbociclib treatment outcome in breast cancer. And then lastly, and this was taken from a presentation at ASMS a few weeks ago, where Michael Fitzgerald's lab at Duke University had used CETSA MS as one of the 3 techniques alongside SPROX and LIP to find prognostic markers for innate resistance in the treatment with oxaliplatin in colorectal cancer. Now, with that, I would like to thank every one of you for listening, and I also owe all of this to the Pelago team at home in Stockholm. And the people involved, they are the ones that have been holding the pipettes to produce this data, and as well as all the help we have from the people running the cell lab facilities and the mass spec facilities. A great thanks to all of you. And then I'm done. I'll take almost any questions.
Thank you very much, Daniel, for that very rich and powerful data you shared. Yes, a blast. So I leave now open for questions. I see directly that we have 2 questions in the chat. And that, are there any special considerations when doing CETSA with monoclonal or antibodies or other biological molecules, or is it generally the same because you are in a low enough temperature range?
Yeah, no, I would say that the antibodies that you add, they're usually quite stable, the monoclonal ones. So that binding will probably be there for quite some time, and it will be, obviously, I think, will be quite large shifts. Considerations is usually, or obviously, it's most often cell surface targets, so you need to have them expressed and so on, which has been tricky in some experiments and in some projects that we have run. But that's otherwise, I think you just have to try it. That's my idea. I think we tried at one point as well to check, and it works, to check the stability of the antibody itself. If you can't access the target protein, you could actually look at the antibody itself and use that as a proxy for the binding, because in reality, it is also a target protein, so to say, as soon as it finds its protein binding partner.
Yeah, so look at the survival of the antibody, you mean, so that is also stabilized, yeah. Another question here, do you always consider destabilizing shifts to be downstream events?
No, the SARC family of kinases is one of the examples that I showed that where this is not. But downstream events, I would say, I don't have the statistics because we haven't no method yet to look into the cell what's really happening. But if I look at a plot with 10 destabilizing hits and 10 stabilizing hits, I'm more likely to say that the 10 stabilized ones have bound to the compound than I would be to say that the 10 destabilized ones had. I think that the destabilized ones are those that are sort of forced out of their original environment, and that is and where there is no net effect of binding something. So if you have a compound that binds to the protein, at least the net effect could be a happy one, so to say.
So if there are no more questions from the audience, I have a couple of questions that I'm curious to hear about, Daniel. So to look at cell type specificities, do you have to use a tool compound, or can you study that on the cells alone?
Yeah, and I think what we do in the Prebiom project that I just flashed is that we look at different donors. We could probably see them as different cell lines if we want, but I wouldn't. Obviously, if you're not adding something, you can't dose it. So you would have to run melt curves So I think that if you run melt curves of different cell lines, you're probably going to end up with a number of proteins that differ in melting temperature. And I think it's going to be more or less, it's not going to be all proteins, but maybe 10% of the proteome that shifts depending on the cell line. That's what I would think. So you run melt curves and look at the melting, the TMs basically of every protein.
Yeah, this is a follow-up question that I came to think of now. So are you surprised over those cell differences that you have seen? Or are you
well, we're not through the data analysis yet. No, I'm not. I think that if the primary target is there, it's very likely that it's going to bind given the concentrations that we add. Yeah, that's but there will be cases like the Sark family of kinases where you have different family members that are important in different cell types. That's one of the known things about Sark family of kinases, is that the family members have different positions or different roles in different cancers.
Yeah. And then so the target engagement atlas is very powerful, and you have profiled now 400 compounds. How will you see it be used in the future? What's the future perspective of the target engagement atlas?
So I think the cell is complex, of course. I don't think that in the near future that we will understand the cell or even a corner of the cell. So it won't be easy to understand why or how things are happening, but I think that we can explain quite a lot of findings in the literature. I think that we can use it to do similarity searches. For example, for our customers, when they have a compound, they send it to us, we profile it alongside other compounds, and for example, we plot it like in one of those t-SNE plots, and you can see more or less if it behaves in a way similar to something else. That could be quite useful. Also, this is the closest that we can get by increasing the size of this atlas. It's the closest thing that we can get to anything near AI, in my opinion. I still think that we would struggle a bit in those kind of powerful applications, because this is data that's where there will be variations depending on lots of factors that are not due to the cell line.
Yeah. Thank you. And then final question. We're approaching the 1 hour here. So as we have the CSO of Pelago and also the co-founder and the one that invented the CETSA method, how do you see the CETSA developed even further in the future? And you know, what do you see for CETSA in the future? It's a big question, but
yeah. Now, when we founded Pelago, we wanted to work quite a lot in the clinic. I see that could happen, or it's happening. I want more of that. So I would very much like to have a CETSA machine at every hospital or every clinical laboratory. Of course, I think that we need to be better at working in small sample sizes and be much more cheap on the cells for the experiments. Otherwise, we won't access primary cell types and this precious material, for example. I think that imaging CETSA could be quite interesting. I know it's hard, but it could still be very interesting. Yeah, and I think that with mass spec, which this was quite a lot about, we are very dependent on the on how the mass spec hardware field is moving, and what we can do with machines in 5 years' time is probably going to be quite different from what we can do now in terms of resolution.
Yeah, thank you, Forward. Looking forward to the future.
Yeah, me too.
So if the audience doesn't have any more questions, I would like to say thank you very much for attending the webinar, and also thank you very much again, Daniel, for presenting this exciting talk to us. So have a nice holiday coming up, and thank you again.
Thank you very much.
Yeah.